Hi, everyone, and welcome to our live interview today. I'm Gabriela, and we will be talking about omics data analysis and results sharing in sequencing units. For this, I'm very happy to introduce you to our two speakers today, Jonathan Landry, bioinformatician at EMBL GeneCore facility, and Murat Akhmedov, who's CEO at Bigomics. Thank you both so much for being here. I will just give a couple of ground rules for our audience. Our interview today will last approximately 30 minutes, of which last few minutes will be dedicated to answering any of your questions. So feel free to drop them in the comment section below. As soon as you have that in mind, we will address them all at the end of the conversation or at the talk. And now I'm happy to leave the stage to our speakers. Murat, you're free to go.
Yeah, thank you, Gabriela. Hi, everyone. It's a pleasure to be meeting you in our next interview sessions. And it's also a pleasure to have Jonathan Landry as a speaker for today's discussion. A quick background information about Jonathan. He completed his PhD at EMBL in genomics and transcriptomics landscape of HeLa cells. He was also a research scientist at Cambridge University. Then he continued as a bioinformatician at EMBL Genome Sequencing Center, in short, GeneCore. Hi, Jonathan. It's a pleasure to have you here. Would you like to provide more information about your background and your current role to our audience today?
Hello, everyone. Thank you, Murat, and thank you, Bigomics team, for hosting me today. Yes, so I'm a bioinformatician at EMBL GeneCore facility. I was a trainer early on as a molecular biologist in France before realizing that working with omics data, I would need a bit more than Excel to analyze those results. And then I moved to EBI in Cambridge, UK. To finish my master. I stayed there for a year and a half, and then before joining EMBL Heidelberg to do my PhD on a genome assembly and also looking at expression profile of HeLa cell line. And after that, I joined the GeneCore facility, still in Germany, to actually support and help users on their projects to analyze the data or train them to analyze their data. Yeah. So that's what I've been doing for the last 9 years almost. Yes. Yeah. And then I, met you on my way, and we discuss about what you are doing, and that's how I'm here. Yeah.
Yeah, thank you. Thank you for your background information. So we would like to a little bit understand how you operate at GeneCore. So would you like to give us a little bit a journey for end users if they would like to sequence at GeneCore? So how would the journey look like?
All right. So we are a sequencing facility. So the main mission of the facility is actually to produce sequencing data. So user groups, research groups are approaching us, having a specific biological questions, and they think that sequencing might help us to understand better their system or to answer their questions. So we sit together, we try to design the experiment. Sometimes they have a precise idea, sometimes they need to have our help. And then they send either ready-to-run libraries, so sequencing libraries that they perform on their own, that they send to the sequencing center to be sequenced, or they send like nucleic acids that we can prepare for the sequencing. We have different methodology, we have different platforms, so we can really adapt the best sequencing strategy to the specific questions. And so at the very then we perform the sequencing, we do some QC, and we might have some requests to actually help to analyze the data. As you can imagine, the range of application could be quite vast, and the range also on the biological question that might be asked is also very different. So depending on the resource that we have, we can address those requests, and then we establish some kind of collaboration. They come in different flavors, of course. It could be like very standard approaches where we do have tool in place. Otherwise, we also can invest a bit of time to actually help them to find a better approach to get like some insight on their question. Yes.
Yeah, it's very interesting. And then in terms of experiments, what kind of data types or experiments your users are mostly generating or running in these years?
So we do have quite a lot of genome assemblies. We also had in terms of type of application, we have a lot of transcriptomic studies. Maybe for the last 5 years, we have like an increase in terms of like single cell approaches, droplet base, also smart-seq, and other kind of approaches, where we see like an appetite for this kind of study. So we are more and more processing this kind of samples. And then we have like ChIP-seq, methyl-seq. So I mean, and you know, we it's not restricted to this couple of methodology that I just cited. It's just like it's very vast. Yeah. So EMBL also like recently has announced this new program which will be focusing on ecology and you know, metagenomics of different environment samples and things like this. So it's really diverse. It's really diverse.
Yeah. And very interesting. And I also heard that your lab is establishing Olink pipelines, right?
Yes. So we do have some pilot experiment on Olink. We actually, I'm not in charge specifically, but there are some people in the lab collaborating with Cellsome GSK that are establishing the methods, and they want to see if this provides like some kind of value to their project.
Yeah, it's a very interesting technology emerging, and it already have been widely used in academic and also industry settings. Yeah. Great. Yeah. Thank you for providing some background about the user journey on data acquisition. Now, my next question is going to be about data analysis. And if we define data analysis in 3 pillars, such as primary, secondary, and tertiary of omics data analysis, where primary is about data acquisition, storage, and management, secondary about pipeline or workflow automation, running mapping, alignment, quantification, and quality control, and tertiary being as data discovery through exploration and visualization, all downstream analysis. So having all these 3 layers of data analysis, to what level would you provide data analysis to your end users?
So on a regular basis, we provide primary and secondary analysis. So we, as I was like saying before, we are a sequencing facility. So we do distribute the sequencing reads at the end. And this comes with some automatic pipeline that look at the quality of those sequences. If we see something you know, weird, we can still discuss with the user if it's relevant or not. We also provide mapping for transcriptomics and quantification. And then for the last pillar, so the discovery or interpretation of results, we are limited resource in terms of bioinformatician in the lab, so we are like only 3, and we, you know, we don't have like all the time to address like or enough resource to address all the requests that we have. So we try to answer the most of them, and also to provide a solution if we don't have enough resource on our side. Yeah, and one is it used to be like also training, so we were hosting for certain period of time the scientist or the biologist in-house, and then we were going through with them with analysis and try to make them, you know, in rhythm, or give them some basic knowledge on how they could start to analyze their data, and then they can build on that later on. Yeah. So the one of the main limitation that we have is this manpower, so to say, and then we try to automatize the way that we do like standard analysis. And recently we start to use more and more your platform on bulk RNA-seq analysis, and try to you know, load the data there and then share the project with directly with the user to enable them to actually browse their own data, and you know, like to guide them through, and if they have questions, they can always come back to us. Yeah.
Yeah, it's very interesting. Yeah, happy to hear that, and that you utilizing Omics Playground for the tertiary analysis aspect. But I just would like to turn back to before Omics Playground. As you mentioned, most of the sequencing units, as well as CROs, provide primary and secondary analysis, and like bandwidth, miss bandwidth in terms of supporting tertiary analysis, or do not support tertiary analysis. So I just would like to highlight here, what are the implications or impact of not being able to provide tertiary analysis for sequencing units?
Well, I mean, like, you know, it's always nice. I guess, like in the team, what like the value that we have is that we can quickly assess if an experiment, like, you know, worked. I mean, like biologists, they have an idea of, like, you know, what they expect to see, what, like if they did like a certain specific experiment, they have knocked out some genes and stuff like this. So they want to have a quick feedback, like, well, is it working or not? And then we can quickly assess that. And also they have, you know, like one solution for everything. Yes. So they send the library, we sequence, we also give them some idea or some interpretation of their with their collaboration on their results, and that's what they like. They have one partner that they can, you know, talk the entire project through. So it's also we are close to the lab, so we know what happened there. We can quickly talk to the wet lab people if there was like some, you know, if they observe some things on the library preparation or the samples quality and things like this. So it's very much like, you know, one team that try to take the project from A and bring it to some stage that is understand, or I mean, it's helpful for the biology, like for the research group. Yeah. So that's what I think what our user likes. And yeah, so we try. I mean, it's very much like a collaborative effort, as you can imagine, like, you know, having this diversity of biological question and also methodology. We really need like some, you know, interaction for us to understand what they are trying to do, or trying, like, what are the questions, how we can best you know, assess this question and answer and potentially answer them. And also we can't be expert like in on all fields. Yeah. So they
Absolutely.
really they really drive the project, and we are here to actually support them in that journey. Yeah.
Mm-hmm. Yeah. Totally makes sense. Totally makes sense. And so, in terms of primary, secondary, and tertiary analysis, so what would be your take on providing end-to-end solution for sequencing units in terms of primary, secondary, and tertiary analysis?
Well, I mean, like, you mean like in terms of like interaction, like, or like project
Interaction,
management?
and then interaction, project management, and also data analysis from the perspective of end users.
Yeah.
So being able to.
Yeah. So basically, what we like very much, especially like on the analysis part, is to actually be involved as early as possible in the project. Yeah. In terms of like design, I think like what is crucial is that we try to design the Rex experiment to answer the question. Yeah. So sometimes we like it happens, I think, like to quite a few analysts where we have some data, but actually there is some biases in terms of in the design or in terms of like the methodology that was used, and then therefore it doesn't answer like very like 100% the question that they were trying to ask. So I think like being involved as early as possible is key. And
Mhm.
also we try to actually, in terms of like having this interaction, so we're trying to actually document what we are doing to that they it's not like we are not providing only plots. We are trying also to explain how we do that,
Mhm.
and why we do that, and things like this, so that they also become familiar with concepts and things like this, and they can potentially recreate those plots on their own. Yeah. So we are
Mhm.
sharing basically all the reports, including scripts and figures that we are producing with
Mhm.
versioning that it's kind of robust.
Mhm.
And recently we started to use your platform because like you also like provide them actually this ability to interact directly with the data. Yeah.
Yes.
So this was like the main, I mean, one of the main feature that for us was quite good is that once the data is in and we drive them through the modules and what your tools is kind of like can do, then they can browse themselves. Yeah. So they can go
Mhm.
interact with the platform, refine the plot, change like the genes, like whatever that they, as they are the expert, they are driving, so they have the expertise, so they know what they want to look at and things like this. So this makes it like very very appealing for us. Yeah. As a
Yeah.
sequencing facility. Yeah.
Yeah, pleasure to hear that. Yeah, you were mentioning that utilizing Omics Playground, especially the complementary part for tertiary analysis, right, and providing end-to-end analysis and to share final results and data with your end users using Omics Playground
Yeah.
for that purpose, right?
Yeah. And I think like we have some kind of understanding what they want to do. So I don't know, we are checking a couple of things on the experiment, like quality wise, and also if the controls are really controls, if they're like the replicates are really replicates, and so on. And we try to extract some meaningful insight, but they like one question brings another one. Yeah. So basically it's always like this um not never ending story, but like you are building your story. Yeah. So you
Yeah.
start with an idea and then you build on. Yeah. So for sequencing facility, and for me personally, it's very nice because like you have always like very like curiosity kicks in, and then you are like also involved in the project. But also on the management side, it's very difficult also to predict how long like a project can we can support a project, because like we have like very creative collaborators. So there are always new ideas and so on. So it's good that at some point they become like also their like like with your platform, with the help with your platform, like some kind of also like the player in the data analysis, and they can look by themselves. Yeah. So that's what is nice about that.
Yeah. Pleasure to hear that. And before Omics Playground, you were saying, how would you sharing your results with the end user?
Yeah, so basically they have access to, of course, like the raw reads, but also like the all intermediate files that we think are relevant, like alignments, and also like any kind of tables or graphics or plots that we are generating. We also provide like them like all the code, like the codes that we are using with versioning. We were simply using like our markdown, and then having like those reports that we are sharing. We were going through them like through this kind of session meeting with them, either remote or in person, and trying to build the story on this interaction. Yes. So this was really like time to discuss the results and to support them in this research project. Yes.
Perfect. And with static reports and, for instance, the static Excel sheets, the interactivity for end user becomes questionable, right? And
yeah, absolutely. I mean, like that's what I like mentioning before, is that after an initial kind of like session with them on your platform, sharing the experiment through your functionality on the platform, we can just like browse the data together, show them how this is actually working, and then they can go on and on their own like check other things or refine the primary analysis to make them like more powerful and more insightful. Yeah.
Yeah, sounds great. And so being a computational biologist, so it's a bit tricky question. What is the difference between a computational biologist and bioinformatician? How would you define that?
Well, that's a good one. I actually never thought about it, but like probably computational biologists might be like someone like more trained in biology, and then use like softwares to actually analyze the data, and probably invest less time in developing the own tool, but like probably using what is already available out there in the literature or in terms of like a tool. And probably bioinformatician, they have this background knowledge on developing their like new approaches in terms of like computational analysis. This might be, I mean, maybe for me, like the slight difference between them. I think that they are both like really, I mean, they are both rare and also like quite important in terms of like, I mean, nowadays I mean, generating sequencing data is not a problem anymore. We do have a lot of platforms that are performing quite well, very powerful, reliable. I think like one of the crucial like bottleneck is making sense of those data. So people. I mean, and it's also on my time. It was like, I mean, I'm not, hopefully not that old, but like the fact that you can manipulate those data and this type of volumes was like necessary. I think nowadays like it becomes like more and more something that the new generation is actually used to and educated, like they receive like education on these topics, so they are more familiar with computer and how to manipulate those data and extract some meaningful information. Yeah.
Yeah, absolutely agree. And in terms of being a computational biologist at EMBL, do you have some tips and advice for other computational biologists or bioinformaticians supporting sequencing units at other places?
Well, I was referring to it like a bit earlier on. Try to be involved, as early as possible in the project. As sometimes, like the it's crucial to have like enough replicates to have enough like depth of sequencing, and maybe like simple design are usually the best to answer like a specific question. So being part of this prior conversation is important, and also I don't know. So probably I think like the what what make like EMBL like a great place to work is the environment. Yes, so we we can interact with a lot of people like having the different field of expertise, different like approaches, different background, and this we can learn a lot from that. So you know. As soon as my knowledge is limited in one field or another, I can just like knock on the next door and get like someone to explain me what's going on. So that's really important, and what I what is it's what I like working at at EMBL is that there are quite good people around me. Yes, so that's good.
Absolutely. Yeah, absolutely. And by the way, you have a fantastic background picture of EMBL. It looks great.
Yeah, I'm my photograph skills are very limited, but we have a very nice photo lab that are providing us like this kind of support. Yeah, so that's good.
Good. I'm jumping in. I see the conversation is very active. I would just want to make sure that if anyone in our audience has any questions for Jonathan, please feel free to leave them in the comment section. But in the meantime, I know Murat has a last question that you have in your mind that you usually keep for last. So if you want to ask that to Jonathan in the meantime, any question you have in mind in the audience, just feel free to put it in the comments.
Yeah, great. My last question would be, so how do you see the future of bioinformatics, or what kind of, let's say, challenges we gonna have, or we heading to?
Okay. So I mean, it's we just have a couple of minutes, I guess. So I need to be focused here. I think one aspect is probably reproducibility and robustness of the analysis. We need to be transparent. We need to document what we do, and we need to be consistent about that. I think we have now great tools that are doing that, and we just need to focus on using them. And in terms of approach, I mean, as I was stating, also we have like increased number of project with single cell application, and it's moving toward now spatial, also transcriptomics. So we are focusing a lot on this kind of approach right now. I think there's great potential to dissect more and more the like like every cell and every kind of subregions of a tissue of a specific biological complex. So basically this is for the like very next future that we have to focus on this. And on the long term, I mean, like more and more we have this, the robotics like AI and also deep learning that are, all around us, and this will also need to be integrated in a sensible way. Yeah. So this is also for later. Yeah.
Absolutely, I agree. Yeah.
Yes.
Thank you so much for sharing your experience, for your insights into the sequencing unit world, which is very interesting, and in general, just for your time today with us. And as our interview is now over, we've reached our time limit. I just want to remind to our audience that if you have any questions or want to learn more about the GeneCore facility, or want to our collaboration with the GeneCore facility, or want to learn more about Omics Playground, you can reach out to our team or just visit our website. You'll find all the information in there, and of course on the EMBL website as well. And yes, thank you all for joining us today, and make sure to follow us on LinkedIn to not miss all of our next events. And thanks again, Jonathan and Murat, for being here today.
Thank you. It was a pleasure. Yeah.
Thank you, Jonathan. Thanks, everyone. Thank you, Gabriela.
Thank you. Bye.